First question was to show the regulation of TRH secretion from TRH neurons. So here's a TRH neuron in the brain. And I wanted a mechanism that is either glucose binding to a glucose receptor, a metabotropic receptor here, and that's what this one shows. Glucose binding to its receptor, metabotropic, which means it's acting through a second messenger. There's the G protein activating adenylate cyclase, giving us cyclic AMP. Cyclic AMP binding to a sodium channel. Sodium comes in, that positive charge starts an action potential. And down here, it opens voltage-gated calcium channels. Should be a calcium plus. Calcium binds to calcium binding protein. That is synaptotagmin. If you want to draw them this way, they're often drawn this way with calcium binding here, binds to a membrane protein. Anyway, that causes fusion and TRH release. If you wanted to show a thermoreceptor, what you'd want is a channel in here. Sorry. If you wanted to show a thermoreceptor, what you'd want is a receptor. Here's a G protein. And in this case, it is one shape when it's warm with a certain number of collisions. And when it cools off with fewer collisions at lower energy, it changes shape, and that causes the G protein to be activated. And here's our G subunit, which can activate adenylate cyclase. This is hypothetical. I don't know if any thermoreceptors actually work that way, but they could. Second was for TSH cell to bind TRH and release TSH. So this particular homework shows the TRH. Here's a close-up showing the events to cause release of TRH. So here's the TRH, and here is the events TRH binding to TSH. And again, this could be a metabotropic receptor up here and causing an action potential. Here it is, TRH binding, opening a sodium channel, causing an action potential, which results in TSH released from the TSH cell. This should not be a neuron. These are in the pituitary gland. They're not neurons. They're just secretory cells. T3 is a major regulator of metabolism. It suppresses some genes and activates others. It tends to activate genes that are important for energy use, and so it increases metabolism. And so we have T3 moving through a transporter into the cell. There's T3 binding to a transcription factor. This person showed T4 not successfully binding to anything. So T4 is the inactive form. T4 actually is weakly active, but T3 is far more active. Anyway, here's our transcription factor. In this case, it's an enhancer for two genes, and so we get the messenger RNA as RNA polymerase moves along, and we get transcription, and it's a repressor for the two other genes. This person just showed one of each. Here is another example, T3 coming in, again through a transporter, binding to the thyroid hormone receptor. The complex then binds here as a transcription factor, genes A and B, it's an enhancer, and gene C and D, it's not. And this person didn't show the RNA polymerase binding moving along and the messenger RNA coming out, but here's the messenger RNA, here's the messenger RNA going through ribosome. Here's our protein coming out, and we get B, and same thing for A. And here is T4 in some area where there's cells that, for whatever reason, need to have a higher metabolism, a higher metabolic rate. T4 in a particular cell near there can be moved into a cell, be converted in an enzyme to T3, take off the correct one of the 4 iodines to make it T3, and released, and now this T3 can affect any nearby cell. So it's only going to be the nearby cells that are higher metabolism. For negative feedback, there are lots of potential mechanisms. This is a reasonable hypothesis. It's quite likely not actually correct, but we could have T4 binding to an ionotropic receptor. If that receptor is a potassium channel, potassium will leave, and that will inhibit the TRH neuron. And this person showed the same thing, T4 binding to a ionotropic receptor that is a potassium channel. Potassium would leave. Again, you would decrease, you'd make the cell more negative and decrease the chance of an action potential. Finally, 5, I wanted a cell that does not express thyroid hormones and how T3 might enter that cell but have no effect. So here's our transporter moving T3 into the cell, but there are no transcription factors expressed here, and so T3 would just be in here for a while and would go back out a transporter. And here's another example just showing, here's the 4 genes we were talking about before. So the enhancer binding sites and repressor binding sites are there, but there's no receptor expressed in this cell. So there's going to be no transcription, no effect.